Good day, Bachelor of Secondary Education, Science majors. I am Wilfred Andrian Villo Obero, and together with Ma'am Aliza Blanca, we are happy to present and discuss to you some of the things that you need to know more about gene expression and protein synthesis. Just listen there, have fun, and as we go through discovering our amazing topics here in biochemistry. So, isn't it amazing? Here in chapter 7, we are going to discuss DNA that leads to RNA and proteins, DNA transcription into RNA, the role of RNA in translation, the genetic code, protein synthesis, gene regulation, mutation, DNA manipulation, and lastly, gene therapy. So, at the end of our discussions, students are expected to answer the learning outcome. First is to define gene expression, describe the function of DNA polymerase, identify the most common sites on tRNA molecule, define genetic code being continuous and unpunctuated, describe the function of protein elongation, differentiate between enhancer and response element, define mutation, and lastly, describe how restrictions in the nucleases operate. So, before we move to our formal discussion, let's define first some of the key terms that we will encounter and are essential for the discussion. We have here the RNA or ribonucleic acids. It is a single-stranded nucleic acid that carries the instructions coded in DNA. We have also here the central dogma of biology or microbiology, where it is the process by which the information in genes flows into protein from DNA to RNA going to production of protein. Polypeptide, when we say polypeptide, it is a chain of amino acids. We have also here codon. The term codon means a sequence of three nucleotides that corresponds with a specific amino acid or start/step signal during translation. We have also here transcription, where it is the process during which a DNA sequence of a gene is copied to make an RNA molecule, while the term translation is the process during which an mRNA molecule is used to assemble amino acids into a polypeptide chains. And lastly, the term mutation, where it is the change in a genetic sequence. Our first topic is the DNA that leads to RNA and protein. So we will questions how the DNA serve as template for RNA and protein synthesis, and the answer is here. So we can see the central dogma of protein synthesis or the central dogma of microbiology. This is the entire process of how the DNA become a template for RNA through the process of transcription, and that RNA are transcribed into a protein through the process of translation, and later on we will discuss each topic thoroughly. To define DNA or deoxyribonucleic acids, it is an organic chemical that contains genetic information and instructions for protein synthesis. DNA structure showing the nucleotide bases or the nitrogenous bases, cytosine, thymine, guanine, and adenine, and linked to a backbone of alternating phosphate and deoxyribose sugar or the S groups. So there are two sugar phosphate chains They are paired through hydrogen bonds. When we say hydrogen bonds, it is usually a weak bonds between the nitrogenous bases between the A and T or adenine and thymine and between guanine and cytosine, thus forming the twin-stranded double helix of the DNA molecule. So basically, deoxyribonucleic acid contains nitrogen bases nucleotide, a carbon phosphate backbone, and a sugar. Um, The configuration of the DNA molecule is highly stable, allowing it to act as a template for the replication of new DNA molecules as well as for the production or the transcription of the related RNA molecule. A segment of DNA that codes for the cell synthesis of a specific protein is what we call gene. The process that lead to DNA to RNA and protein is what we call gene expression. Gene expressions are also called protein synthesis or transcription or translation. Gene expression. So what is gene expression? Gene's expression is the process by which information from gene is used in the synthesis of a functional gene product that enables it to produce end products. So protein or non-coding RNA and ultimately affect the phenotype as its final effect. So, what is the role of DNA polymerase? The primary role of DNA polymerase is to accurately and efficiently replicate the genome in order to ensure the maintenance of genetic information and its faithful transmission through generations.